This study was about asking the question, what's the difference if we react instantaneously or if we plan an action, can prepare for it and react when we fully planned. A good example would be, for example, you sitting on a start line for a sprint and waiting for the go signal, you know will appear in 10 seconds. Or in contrast, you're already running full speed in the race and your opponent just crashed down and you have to quickly jump over him. So the first one would be a planned movement. The second one, in contrast, would be an instantaneous movement because you have to, as quickly as possible, jump aside. So in order to investigate this, we trained 2 macaque monkeys to perform a task, a grasping task. So we were looking for grasp movements. And to give an idea, monkeys were sitting in front of a grasping box. What is a grasping box? It has a handle in front. And monkeys were trained first to grasp this box with either a power grip or a precision grip. To contrast delayed and instantaneous movements, there was an additional cue given to the monkey which told the monkey when to react, when to grasp. And we not just had one condition, we have plenty of them, starting from instantaneous 0 millisecond time to 1.3 seconds or 1,300 milliseconds. And we had plenty of conditions in between to really investigate the continuum between instantaneous and delayed grasp movements. Why did we use macaque monkeys? The reason was simply we wanted to understand the brain activity going along with it. And unfortunately, up to today, the only way to really investigate neuronal activity of individual neurons is possible with invasive methods such as electrophysiology. How can you imagine that? So we were interested in the brain. Here's front. Here's back. And we know already from plenty of studies, movements are basically controlled here in this region. Also hand movements. So what did we do? We implanted chronic floating arrays. These are tiny, tiny devices, few millimeters in size, with small electrodes sticking out. To give an idea of an electrode is thinner than a human hair. And the tip is even not visible. It's on microscopic scale. And we implanted a couple of these in different grasp areas and asked the question, how is really this continuum encoded in these brain areas relevant for grasping? The result was, first to say, was the whole population across both areas encoding these processes. But to give an example, this is now just for the whole hand grasp. And what you see here is now from dark blue to light blue, the different conditions I showed here. So 0 milliseconds to 1.3 milliseconds. And what you see here is now the population activity of all neurons aligned to the initiation of the movement. And the interesting finding was that in the time before, that is 200 milliseconds prior to the movement initiation, there were 2 different states. One for really immediate, instantaneous movements. So when the monkey had no time to wait to memorize it, and a different state for movements out of memory. And they were really distinct and separatable. And that was the case for both grip types. I mean, why is this relevant? The long-term goal here at the DPZ of our research group is motor prosthesis. So the ideal goal would be to really allow tetraplegic patients to move again. And these implants we use are the implants used to get the signal to move robot arms with, of course, a big computer in between, and there comes the problem in. It's a lot of calculation time you need for first encoding the signals and then transfer it into trajectories for robot arm control. And so time is a crucial factor, and our attempt here is to basically use the activity prior to the movement to already let the computer calculate something which can just be initialized when the patient wants to move. Unfortunately, I mean, lucky for us, it ended up in a study, but unfortunately, this is not that easy, because if you have something in memory or react instantaneously, you are in a completely different state. However, if thanks to our study, if you consider that, you can still make use of it and still use it for presumably motor prosthesis in the future for artificial limbs.